Hello everyone, this is Rajamanikam Antonimuthu from QPT. A team of engineers led by the University of Massachusetts Amherst and including colleagues from the Massachusetts Institute of Technology MIT recently announced in the journal Nature Communications that they had successfully built a tissue-like bioelectronic mesh system integrated with an array of atom-thin graphene sensors that can simultaneously measure both the electrical signal and the physical movement of cells in lab-grown human cardiac tissue. In a research first, this tissue-like mesh can grow along with the cardiac cells, allowing researchers to observe how the heart's mechanical and electrical functions change during the developmental process. The new device is a boon for those studying cardiac disease as well as those studying the potentially toxic side effects of many common drug therapies. Cardiac disease is the leading cause of human morbidity and mortality across the world. The heart is also very sensitive to the therapeutic drugs, and the pharmaceutical industry spends millions of dollars in testing to make sure that its products are safe. However, ways to effectively monitor living cardiac tissue are extremely limited. In part, this is because it is very risky to implant sensors in a living heart, but also because the heart is a complex kind of muscle with more than one thing that needs monitoring. Cardiac tissue is very special. It has a mechanical activity, the contractions and relaxations that pump blood through our body, coupled to an electrical signal that controls that activity. But today's sensors can typically only measure one characteristic at a time, and a two-sensor device that could measure both charge and movement would be so bulky as to impede the cardiac tissue's function. Until now, there was no single sensor capable of measuring the heart's dual properties without interfering with its functioning. The new device is built of two critical components. The first is a three-dimensional cardiac microtissue (CMT) grown in a lab from human stem cells. CMT has become the preferred model for in vitro testing because it is the closest analog to a full-size living human heart. However, because CMT is grown in a test tube, it has to mature, a process that takes time and can easily be disrupted by a clumsy sensor. The second critical component involves graphene, a pure carbon substance only one atom thick. Graphene has a few surprising quirks to its nature that make it perfect for a cardiac sensor. Graphene is electrically conductive, and so it can sense the electrical charges shooting through cardiac tissue. It is also piezoresistive, which means that as it is touched, say by the beating of your heart, its electrical resistance increases. And because graphene is impossibly thin, it can register even the tiniest flutter of muscle contraction or relaxation and can do so without impeding the heart's function, all through the maturation process. The researchers then embedded a series of graphene sensors in a soft, stretchable, porous mesh scaffold they developed that has close structural and mechanical properties to human tissue and which can be applied non-invasively to cardiac tissue. Graphene can survive in a biological environment without degrading for a very long time and not lose its conductivity, so we can monitor the CMT across its entire maturation process. The researcher says that their sensor can give real-time feedback to scientists and drug researchers, and it can do so in a cost-effective way. Thanks for watching this video. Now you can get my ebooks with a huge discount at www.rajamanikam.com by using the discount code QPT. Contact me if you are interested in earning a 50% commission for selling my ebooks and course. Thanks.